All right, guys, let's start 1.3 notes membrane structure. This is a very notes heavy part of the chapter, so this might take a couple of screencastifys. The understandings for this chapter: phospholipids form bilayers in water due to the amphipathic properties of phospholipid molecules. Membrane proteins are diverse in terms of structure, position in the membrane, and function. And finally, cholesterol is a component of animal cell membranes. All right, so let's start with phospholipid bilayers. The word hydrophilic means attracted to water. Hydrophobic means it repels water. Phospholipids, which I'm going to be abbreviating as PLs, have parts that are both. This is called amphipathic, like amphibian. It's both water and air. Amphipathic means both hydrophilic and hydrophobic. The phosphate head is hydrophilic. The fatty acid or hydrocarbon tails are hydrophobic. We'll see pictures of these in a bit. Exposed to waters, the phosphate groups are attracted to water, but the tails are attracted to each other, forming a bilayer. This way, the phospholipid heads are in the water, basically, and the tails are repelling water by facing each other. So here we have a picture of, on the left is a phospholipid. The hydrophilic head is that phosphate group, and then it's got two hydrophobic tails, and the bilayer sheet is what you see there on the bottom right. When exposed to water, so the water is above and below the membrane with the phosphate heads in that water, and then the tails forming a hydrophobic or like an oily kind of center. So let's look at some membrane models. In the 20s, Gorder and Grendel deduced that phospholipids were arranged in a bilayer, but their model did not explain where the membrane proteins were. So this section's all about trying to find out where those membrane proteins are. In the 30s, Davison and Danieli proposed layers of protein on both sides of the membrane, a sandwich model. So you've got the phospholipid bilayer, and then you've just got a layer of protein on top and below on those phosphate heads. Then in the 15s, electron, that E negative, because they're negatively charged, that is my symbol for electrons. Electron micrographs showed a railroad track appearance, dark protein lines with a lighter phospholipid band between, kind of fitting the Davison and Danieli model. But then in 1966, Singer and Nicholson proposed peripheral proteins attached on inner and outer layers. Integral proteins are embedded. This is the fluid mosaic model of today, and we'll see some pictures of these. Here's the Davison-Danieli model from 1935. The sandwich model, you can see the purple are the protein layers on either side of the phospholipid bilayer. Problems with the Davison-Danieli model. They did freeze-etched electron micrographs. This is where cells were frozen and fractured along the center of membranes. And they had globular structures that were interpreted as transmembrane proteins. Transmembrane means the proteins are going through the membrane, not just on top or bottom of the membrane. Then the structure of membrane proteins. Proteins were extracted from membranes, and they varied in size, and they were globular, unlike the type that would form a continuous layer on the periphery. Also, proteins were partially hydrophobic, so they would be attracted to the hydrocarbon tails of the phospholipids, so that they would be transmembrane, going through the membrane. Finally, fluorescent antibody tagging. Membrane proteins were tagged with green labels on some cells and red on the others. The cells were fused together. The red and green markers eventually mixed, showing membrane proteins are free to move within the membrane rather than be fixed in the peripheral layer. All right, so it's going to cut me off before I can get through here. So I will start talking about the function of membrane proteins, but I only have a few seconds left.